So this is a very big day for anyone who has cardiovascular heart disease, or anyone really who loves somebody who does, because the One Brave Idea Science Innovation Center opened yesterday. Yay! Very big. Awesome. And it represents the American Heart Association, Verily, and AstraZeneca working together to bring an end to coronary heart disease, with a goal of, in the process, and I love this part, uncovering foundationally new approaches to wellness and eradicating disease that extend across an even broader definition of health. And I think that's incredibly exciting. So the center is going to house the $75 million research enterprises leader, Dr. Callum McRae, who is the chief of cardiovascular medicine at Brigham and Women's Hospital on the cardiovascular medicine team, and his world-renowned team of investigators across a range of different institutions and different disciplines. So you can think of the center really as the hub for all the work that's being conducted right now to identify the earliest markers of heart disease and to develop strategies to stop the disease before any health crisis has a chance to come to fruition. So this is really about bringing together the best and the brightest from the areas that I think you would expect, but also, and for me much more interestingly, maybe from some that you might not. And we're going to talk about that more with you. I also think, and I'm going to use these as my words and I won't put these on anyone else, but this is about creating a safe space for failure. Because solving this problem is going to require new collaborations and approaches that have not been done before, and therefore, by definition, failure will be part of that process. And one where what's not working is as informative, ultimately, as the stuff that does, with the outcome obviously being new prevention and treatment strategies. So this is really an incredible example of 1 and 1 and 1 adding up to 10. So you've got that unique mind meld of AstraZeneca's proprietary data with their existing and novel data sets. You've got Verily's informatic capabilities and their ability to innovate. And you've got the American Heart Association's already unparalleled and ever-evolving ecosystem of patient and human-centered research and scientific networks, all really coming together to pursue one vision, one brave idea, under Callum's guidance, and heart, and genius, and leadership. So 2 other quick thoughts I have to share. Anyone who is in marketing, and not enough of us in the healthcare space are in marketing, but let's all remind ourselves we're marketers on some level, knows how much words matter. And I love the name of your center, One Brave Idea. And I think it's telling because it's about making progress happen. And the word idea does not just reflect, as I suspected, just a thought or a plan, but the definition of idea, if you look it up, is a plan for action. And the only way change happens, as all of us know, is through action. It's a beautiful thing. And often, almost always, that action will be hard. And that's why I also love the use of the word brave being front and center here. Because as a bootstrapper, as an entrepreneur, what comes into your head when you think of the word brave, right? You think of like hunkering down, you've got a shield, you're going to battle, but you're going to battle about something that really, really matters, that's worth that extraordinary effort that you're going to give. And I think cardiovascular health is worth fighting for. So congratulations on the launch of your new center. And I want to stay with the idea of definitions for a second, because I think it's central to part of the real beauty of what you are doing here. And in our conversation, we had a great conversation. May all of you have a chance to have a conversation with Callum tonight. In our conversation, you made the distinction between understanding disease better versus understanding what it is to be healthy. Tell me more what you mean about that.
Well, Alex, thanks first of all for that introduction. I think I prefer to focus on the fact that I am indeed an expert in failure. I think the One Brave Idea was actually the title of the original application, and I think the bravest thing was having a really transformative approach to how the applications were assessed and awarded. I think that itself was an amazing experience, learning how to work with other people as we went through the process. I think that was incredible. But to get back to your question, I think, and actually I think Fred and Deb brought this up, as did the last question. I think the core problem in much of medicine is information content. And I think almost all of what we focus on is really largely late in disease. We wait for people to come to us with a problem. And that's been quite successful up to a point. But I think it does narrow the scope of how we think about the biology that ultimately is the only real constant in medicine. And so the goal of the program is really to try and move the needle back to build on what's gone before. And cardiology, I think, has been a remarkable pioneer in medical fields in this setting, and has always tried to move earlier and earlier in disease. And what we've done is really just take that and make a natural extension that's now feasible because of what we know in biology, what's possible in technology, and what we need to do in order to enrich the information content in medicine. And that's to move back and to begin to really understand that at a cellular and molecular and social and psychological and behavioral manner, what's really going on at that boundary between when you're last well and you first start to drift towards what ultimately is still the most common cause of death in the developed world, atherosclerosis and its downstream consequences.
So tell me a little bit more about this notion of detecting it early on before it's disease. And some of the language I've heard used is an early warning system and a sixth sense. Yeah,
I think so. I mean, a lot of these things are ways of capturing ideas that ultimately are really very granular approaches to trying to understand what's going wrong when it first goes wrong. I think many diseases, and we heard some of this discussed even this evening, are really aggregates of multiple different things. They're probably about, we know already, at least 30 or 40 conditions that cause diabetes. There are probably at least as many core conditions that cause coronary heart disease. And what we've done is, I think, not deliberately, but simply because of the burden of disease, focused on those later stages when everything really is in a final common pathway. And what we need to do is to move earlier and earlier so that we can actually stratify not imperfectly late, but actually very precisely early. And by focusing on what the actual fundamental biology really is. And actually, one of the things that's quite interesting to me in medicine is that and I see this happening around us, is what we're doing is we're digitizing the 18th century or the 19th century at a push. And we're not actually asking ourselves, what really could we do differently? How could we change the information that we actually gather rather than just gathering more of what we've already gathered for the last 20 or 30 years? How could we begin to analyze it differently? We heard a discussion of that. But also, how can we begin to think, where are the gaps in the information that we need to fill, and how can we, in a prospective and proactive fashion, build tools to actually understand that, build those tools deliberately in a fashion that's scalable, and then begin to integrate with the existing information and some of the data that we are now able to collect, whether it's behavioral information from our mobile devices, or whether it's something that's happening in our home, or even some simple other ambient sensors that you can imagine, whether in your car or your living room, or just even your television behaviors. Those types of things end up, I think, only aggregating to inform biology if we understand more about the core biology. And medicine, I think, is also one of the few areas where the research and development happens in a different industry than the actual implementation. They're like two separate spaces. And I think the thing that, from my standpoint, has been most exciting watching digitization begin to happen in medicine is that for the first time we realize we can't afford to have two separate systems. Mm-hmm. But ultimately, many of the things we're recording are things we have 30 years worth of data on. Mm-hmm. And we know already they don't really help us discriminate that effectively. And so by understanding, I think at a more granular level, where the gaps are in our knowledge, and much of that means going back to times in the disease process when you can still discriminate the difference between the different disease types, that we'll be able to understand it better in the future. So one of the things you said during our conversation was your heart is not just about your heart, it's about your whole body. Absolutely. And I think that that's a natural consequence of us waiting until a organ-specific problem arises. But we all know, everybody who has the disease, even the patients themselves, realize this is a systemic disease. And I think we heard, again, even in the last session, how medicine is, as a enterprise is not really built for as a system. But much more importantly, biology is nowhere near to understanding our bodies as a system. What we've tended to do is focus on the anatomical or physiologic findings that first appeared when the diseases emerged. And we haven't really moved back from that in a progressive or in as radical a way as I think we need to say, what is it at a fundamental level that's different, and how can we first detect it? So many of the ways that we think about even conceiving it are improving the technologies that already exist rather than moving outside the system. And that's another thing that I think patients are very much better at doing than we are. Systems are very much better at doing than we are, which is collecting data whether we want the data to actually appear or not. It's going to come in, and by taking an agnostic approach to analyzing whether it's nerve function in a cardiac patient or cardiac function in a nerve patient, or behavior, or taste preference, or olfaction, or mood correlations with your oxygen saturation. Those types of things ultimately are the types of biological processes that are actually, we know already, fundamentally linked to the underlying core mechanisms of at least some forms of coronary disease. We haven't really tried to use that to dissect it. We have a very medical model for thinking about how we're going to...
Well, and something we had talked about, so relatives can often tell when a member of a younger generation seems likely to develop the same condition. And there was an example you gave where children with an underlying form of heart disease, you noticed something you weren't looking for around their balance.
Yeah,
so that was a great example.
Again, this is a classic thing that obviously geneticists will do, is if you're trying to understand what a disease looks like. So when genetics, one of the best things about genetics is you see the progress of disease through multiple generations, so you understand how it can eventually manifest. What we haven't done enough, I think, is go back in time and actually ask children, what's the first thing that you noticed was different? And so we did this actually with a rare form of arterial blockage, and we said, what are the things that you noticed that were different from your siblings? And they were actually not even cardiovascular, as you alluded to. They were loss of balance when playing sports compared with their siblings, and also a tendency for their legs to become numb more quickly when they were sitting cross-legged. And that information literally led us to begin to explore nerve function, which, again, we've probably known. If you look at the biology of the genes that cause coronary disease, it's been known for 20 or 30 years that it affects synapse development. It affects every membrane in every cell in your body. But we've never really moved outside the arterial tree, or at least the vascular tree, to try and understand it. And turns out these manifestations are present maybe decades before the actual arterial blockages occur, at least in this very small subset. So can we begin to, by thinking more broadly and by actually making directed efforts to discover completely new phenotypes, new data streams, can we actually begin to extend the repertoire of what we consider as biologic data? And importantly, as was discussed in the last forum, think really about the information content. You know, one of the things that always amazes me is we've recorded intracardiac data beat by beat from tens of millions of people in the highest risk populations, and we still haven't been able to infer from that data who's actually going to have an event in a way that meaningfully affects the individual patient. And so that tells us that there's just not enough information in the data that we're actually collecting. And so by broadening dramatically the type of information that we consider medical, and by actively expanding that repertoire, I think we're going to have not only early warning systems for disease, new ways of detecting whether your cell metabolism is working, whether you're handling lipids normally, whether your inflammatory cells are working, not by the age of 40 or 50, but by the age of 2 or 3 or 5 or 15. When we know already the disease can be detected, but we just haven't gone out looking for it, then I think we're going to really enrich how AI and other technologies that have had, I think, a very deep role in some industries, largely industries where the information content of the data that is being analyzed is high, not where we've sort of sat in a silo that was established by people somewhere around 100 years ago, and just we dug deeper and deeper in that one area rather than moving outside.
Well, what you're talking about on some level is we want to look back as far as we can for something that we don't know what we're looking for.
Exactly.
So how do you do that with order, and how do you bring the ability to then, go through that data and derive meaning, look for signal in noise?
So that's actually, I think, something we don't really know how to do, and that's partly what I think is the mission of One Brave Idea, is to begin to think systematically about how you can expand that phenotypic spectrum, that set of tools that you might use, in at least one diagnostic category, one biological process. I think the way that we've thought about it is there are essentially two strategies. One is to use information that already exists and use it more rationally. And when I say rationally, I'm thinking about it not being in any way critical of what's being done up until now, but thinking about it in terms of how would you add value to those data that would inform a system. And oftentimes that's challenging the system. And then the second approach that we would say we should take is to think about what are the biological classifiers, how can we test in multiple different areas, knowing that many of them will fail, whether those biological classifiers actually allow us to detect a signal that's relevant to this disease. So what do I mean? So you might imagine, obviously, you would look at a particular biological process that has already been implicated in coronary disease. So every gene that's already known to definitively cause coronary disease affects a process called endocytosis, where the membrane takes stuff from the outside and moves it in. Nobody's ever thought of a way of measuring that at scale. Can we do that? Well, we'll test it and see. But there are lots of other areas that are equally important. How you use energy. Can we measure that in people in a way? Do they use fatty acids? Do they use carbohydrates? How do they metabolize those things? Can we characterize that early enough to be able to understand, can we then actually modify how people think about their nutrition? And then that gets actually to another major component of this, which is we've known also for a long time that coronary disease is really also a function of a very powerful interaction between genes and environment, but we don't measure enough in the environment to really understand what are the true triggers, or are the triggers in some people and not in others. And so I think one of the most exciting projects is one led by one of the team members, Laszlo Barabashi at Northeastern, looking at really trying to characterize, not in terms of the 150 USDA constituents of food, but actually in terms of tens of thousands of molecular constituents. What are the differences in exposure between the people who develop disease and the people that don't? We couldn't do that 20 years ago because we couldn't document, or even 5 years ago, 3 years ago, document at an individual basis what everybody's actually eating and then map that back onto a database. So by connecting Laszlo's analytics skills with existing biochemical databases that are evolving and thinking about working with Verily and how to capture information on food as it's being eaten, as it's being consumed, and put that all together, we hope that we'll be able to understand at least how we might characterize what the environmental factors are at a more granular level. So it's the same basic premise that I mentioned when we're thinking about what we measure in the body. Is we've tended to settle for not through anything other than because it was necessary, but we've tended to end up with a fixed set of data that we have worked with again and again, and we've extracted a lot of the information from that. And by moving outside into spaces which are much less comfortable, much less fully medicalized, much more amenable to confounding, we begin to both expose ourselves to the risk of failure, but also try and find the ways forward that we believe might be useful not only for coronary disease but for other diseases.
So I would love to stick with failure for a second, because I sat here I had two emotions as you were describing this, like, wow, that is just the coolest thing ever, and then it's just exhaustion and thinking about how much there is that has to get done. So I want to read a quote, because I think built into the essence of what you're doing is working through failure productively. And you got to try things categorically that don't work. And so the CMO of Verily, Jessica Mega, said, "This model allows our investigators to hit roadblocks, reevaluate, and move on quickly. Every effort will provide meaningful information, even if the answer is to look someplace else." So my question for you, and I actually have double fun asking this because I think one of the things Walmart has worked on since the early days of Sam Walton is how do you try and fail fast? So how are you building a culture that really allows for this, especially in an industry that seems to want to hide failure?
That's a very important point. I think what we're doing, and we're obviously feeling our way through this in real time, is we're trying to make sure that we're very goal directed. This is a substantial amount of money. There's a very clear goal. One of the biggest problems with biology is it's almost so expansive, as you mentioned a minute ago, that you can't put your arms around it. In fact, one of the most interesting things, I've said this for a couple of years, but the only bounded data set in all of biology is the genome. That's why people cling. I'm a geneticist, so I like clinging on to genetics, but that's why people cling on to it, because it is truly the only bounded data set. And the difficulty is that even the scale of the other data sets is an unknown. Yeah. And so what we're trying to do is make sure that we keep our eye on the prize, on where we're headed. And so we're trying to be creative enough in the initial hypotheses that we set, that at the fundamental first step is understanding whether or not the results from that project will inform coronary disease. So we're trying to very quickly, very early in every project, understand, does this or does this not have enough signal to impact coronary disease, at least as we can best detect? And we know we're going to have both false positives and false negatives. There'll be some things that we may think have failed now, and we move on, and we may have tested, may have even failed in testing the failure. We recognize that. But one of the things that's important about screening, and this is something I've learned through many years of doing high throughput screening for hundreds of thousands of variables, is that what you have to go with are the positives that push you in the right direction. And so by testing enough things in a careful enough manner, in a well enough classified manner up front, you can begin to narrow in on areas that you know are positive and are heading in the right direction. And you're not assuming that the other areas are truly negative. You're just assuming they're not where the biggest value is at this moment in time. And I think that's something that we don't do very well in biological research. We tend to hold on to an area. I was thinking back to what it was that sort of changed my mind about this, and it was reading Karl Popper's Conjecture and Refutations. I think we always imagine that the real important thing in biology is the original hypothesis. But actually it's designing the hypothesis so that it can be refuted. And what we tend to do is we tend to design our hypotheses so they can be partially refuted so we can continue to work in that space. Yeah. And by not being rigorous enough in how we define what the question really is up front, we end up limping along, and I've done it as much as anybody. But it's something that biologists do in general because of the fact that biology is so complicated. This is not a criticism; it's an observation of something that we've all done, and it's because you can't frame the hypotheses clearly enough sometimes, because there's so many unknown unknowns. We tend to go, well, there was this thing that we didn't think about. Let's go again along this exact same route, but with a very subtle extra step. And I think we need to say in many ways, where is the signal? Let's follow the signal, and then come back in the future to areas that weren't productive, not throw them away, but learning, how to prioritize things is something that many other scientific areas have done very well for a long time. And that's partly because they've recognized how important team science is.
So I'm thinking about how everyone in the audience, I always, when I listen to someone, think like, how do I apply this? How do I apply this? What does this mean for me? Which might make you think I'm very self-centered, hopefully not. But there's a Mark Twain, my favorite quote is, it would have been shorter if I had more time.
Yes.
And you mentioned before the process for submitting to get chosen. And I think one of the examples I read about you is that you had submitted on for one grant a 439-page thing, and your grant, one of the things that's so unique, huge kudos to you, about this process was how short it was, that you actually you were forced to come down to 250-word responses, which forces a clarity in what are your goals, what are you really trying to do? Tell us a little about that process.
Absolutely. So I think that really was the brave part of the whole idea, was actually I think it was Nancy and Andy Conrad, and I think Bob Harrington was in the room as well, who really thought about how do you make this a different process from start to finish. So having a 250-word primary idea as the initial screen, I think was really, it was particularly attractive to me. Because as you can tell, the longer I go on, the less coherent I become. But the fundamental element that was important was that it really changed the way that we think about how we communicate, not only to the funders, but also to the people around us, what the ideas are. And so that for me was, I think, a powerful organizing principle, which, as you can tell, I've allowed partially to lapse, which was being very concise and direct. But I think one of the things we recognize that to be true to the vision that the AHA, Verily, and AstraZeneca brought to the table, we really do need to be able to frame what we're doing so coherently that we are actually testing it in a thumbs up, thumbs down fashion up front.
Well, and that's the process, right? If you got to try a lot of stuff, fail fast, so that you can continue to get that clarity.
To get that clarity.
So key to that also is a team.
Yes.
So you've got to have extraordinary people that you're working with. Give us some examples of the characteristics that you're looking for, and some examples of unusual folks who you have on your team that we might not have expected.
So I think one of the things that we were definitely looking for were people who cross boundaries, and I think that's something that was, for us, vitally important. People who had a history, actually, to be honest, not just of crossing boundaries, but also of going into a field, recognizing the difficulties and moving into another field to solve the same problem. And so some of the individuals really have remarkable expertise in different areas. We're fortunate to have, for example, I mentioned Laszlo. We also have, who's worked in network science across multiple different scientific disciplines. We have a geneticist and a fellow Scott, Ewan Ashley in Stanford, who has really been transformative at the boundary between whole genomes and understanding how to interpret them. We have a yeast geneticist in Toronto who's literally only worked on yeast for the last 20 years, who's really thinking about how the biology of a single cell organism can inform the complexity of a whole human. And I think there are a lot of really remarkable analogies. If you think about it from an evolutionary standpoint, at least 80% of the evolution that occurred in biology occurred before two cells came together. So that's actually an important thing for us to think about. A lot of the fundamental mechanics are really dependent. You need those fundamental mechanics just to form one cell. So what can go wrong once you get beyond that point is really a function of what's happened before. That's, I think, a good theme for moving everything earlier. We have a venture capitalist who has a track record of actually building virtual companies, of taking things out and instead of setting up in traditional fashion a startup, of actually building, using pieces of other companies, a virtual company that he can then use knitting things together to execute in a particular space that then allows him to pick a direction to take that company in, and then he founds the company. So really doing empiric experimentation early on is a core part of this. We also have, for example, Elazar Edelman, who is one of the most senior bioengineers at MIT and also works at Brigham and Women's Hospital. He was an integral part of inventing the stent, something that is already critically important in our current framework of current disease. So we did not want to throw away the past, but we wanted somebody who has a track record of finding a problem, building a solution, and reducing it to practice. And that ultimately is also, I think, a core competency that we really wanted to make sure we brought into the team, the ability to actually move things to practice, not to be theoretical only, but to test that theory, and then once you have data that suggests that it is worth moving forward, to move it forward in real time. I think that's critical.
So finding opportunities for folks to work together in different ways and how they communicate with each other. I have two very quick questions on that. Number one, what does your space physically look like, and how are you making sure that you're throwing off the feeling of being part of a traditional hospital or a traditional, more traditional clinical center?
So we've deliberately moved the space, the One Brave Idea Innovation Center, just away from the hospital, far enough to be discreet, but we've designed it like a startup company, essentially. We've made it look like as much like Google as we could dare to do without breaching copyright. But no, we've literally broken down walls. We've made it intensely collaborative. When I think Nancy and Bob and I were sitting at coffee one day just thinking about it, and the concept of an incubator, that sense of actually having young ideas grow up and merge was something that we really wanted to foster, and so that's how we've designed the space. The other thing that's important is to recognize that this is the hub of an ecosystem that we believe will link out. What I don't want this to become is a single center. I mentioned already, we have direct collaborations as part of the original founding team with Toronto, with Northwestern, with Stanford, with Northeastern. What we want to do is to also bring people in as we need them to solve the problem. This is a dynamic team. This is not a static, you're moving forward as a single large mass. We are basically trying to work out who do we need to solve this particular problem, and once we have solved that problem, maybe those people will adopt an advisory role, we'll bring somebody else in to drive the next step.
Yeah.
A lot of this is special teams and trying to understand how to bring people together across the country and across the world to leverage the resources we've been given to solve that problem. And if that means going to industry to get the expertise, we will do that. One of the really important things about the whole project is it is being run like a not-for-profit venture, and we have a board. We have the funders actively involved on a quarterly basis and more continuously on individual projects in helping us think through what are the best ways of moving forward. And that for me is a particularly amazing experience, to have the expertise that's present on the board and that we can bring in a cyclical basis for other problems. That luxury is really important, I think, for all of us.
And I think just to continue with this idea of trying to break down old ways and old constructs, I read someplace, Nancy, that the way you even interact with the board is different. So there are not long memos, you're not sending 30-page reports, that there's much more dynamic interaction.
Very much so. It's literally an executive summary. We're asking the board for input on key decisions. We're presenting the data to allow them to make informed choices on those key decisions, or make informed advice on those key decisions.
Yeah.
That's essentially how it operates.
So there's a theme with you always, I'm hearing of like progress, right? How do we keep moving? How do we keep learning? Building something sustainable and scalable, and being shameless really in your pursuit of whatever it takes to get that done. There's the old adage, you can't manage what you don't measure.
Yes.
And you said specifically, but if all we're doing is just measuring better, but measuring the same things, we won't see improvement. In this, you're measuring yourself all the time. You're measuring the progress that you're making. Talk to us about some of the things that you're recognizing that have not been measured, that you're bringing discipline around measuring.
Well, so one of the things, as you can tell, that's probably a bugbear of mine, is that we don't measure the responses to drugs. If you think about the challenges that you can use in biology that allow you to integrate from a single molecule to a population, there are really only two things that operate at those scales, and that's a biophysical challenge and a small molecule challenge. Medicine is the single largest small molecule operation on the planet, and we essentially don't measure a single response, with a couple of notable exceptions. And so part of what we imagine is if we can identify the right things to measure, and we layer in on top of that the challenges that we already use, and in many ways, some of that may be exactly what we're going to do, is take existing technologies and just look at them before and after a challenge to the system that we know is important already in that pathway. We're changing the dynamic range of the measurements we make. We're changing our understanding of how robust the system is. And we're beginning to use data that we should have been collecting all along, but have been throwing out.
Yeah.
So one of the most amazing things to me, I was listening to the discussion earlier, we've been building sort of, one of the things that we did realize is physicians are part of the problem. So we built some implementation programs that eliminate doctors. And one of the things that we recognized was that the information is actually, it's not just siloed, it's not even remotely connected, and it's not collected in ways that are relevant. So in many ways, the pharmacy has data on prescriptions, the pairs of data on claims, but nobody's ever really truly worked out what the fundamental biological relationships are between those two data sets. And actually the patients are a really important source of how to link those things in real time. And so just reinventing how we imagine the workflow of the data we collect. Those data are great if you collect them at the right metadata. We throw away most of the metadata or we never collect it. Understanding how we can just take what we're doing already, refashion it in a way where we collect the information at the right time, in the right place, with the right stimulus. And then suddenly we've got a whole new layer of data before we add a single new measurement. Once you start then to add measurements to that portfolio, you're beginning to really enrich the data sets that you collect. You can then begin to understand the genomes that you've already sequenced. The rate-limiting step in all of genetics is phenotype, and that's partly because we measure so few phenotypes. If you add every single thing in the entire human phenotype ontology together, it comes to about 9,800 variables. So it's hardly surprising you're not going to be able to deconvolute a genome that is 5 log orders more complex than that. And that's just the base structure, far less all the downstream modifications of RNA and proteins and cell biology that accumulate over time. So we need to really start to think about how we enrich the information that we're collecting.
One of the things that we've talked about, I'm a huge believer in this, is a lot of the things that as an industry we've historically thought of as being soft data.
Yes.
We're actually coming to realize is maybe really the hard data, and it's the stuff that we should be capturing. And this word ontology, and how do you organize data? Can you just give a sense quickly, an example of unusual bits of data that you are capturing in a way that is allowing you to build these structures?
Well, so obviously there are already, I think, fairly robust ontologies that are very much driven by how diseases were conceived of and evolved. And so one of the things that we're thinking about is taking ontologies that may already exist, as I mentioned, in, let's say, cell biology, and moving those into the clinic. So that's work that I think we could very quickly do and work we've already started. But even thinking about how you classify disease based not on the end stages but on the early stages, that itself requires a completely new ontology because we don't know all the structured steps that link, let's say, a developmental abnormality to the final blockage in the artery 50 years later. So we need to fill out those steps. One of the areas that we've really begun to focus on quite a lot in the broader sense is thinking what are the areas where ontologies are truly necessary. I think symptoms is one area. If you think about what is the difference between us and any of the models that we might study, whether cellular or molecular or animal models, it's the ability to converse with the patient. And if we throw away that information, as I mentioned earlier, when talking to the children who have inherited rare diseases, we throw away the information that they bring to us, we're really throwing away the most valuable parts of phenotyping that we have unique access to in humans. And so one of the things that we're actually thinking about doing and have already begun to explore is actually using children to define wellness. So actually getting back and saying, we define wellness in terms of what we're doing as adults, but maybe that itself is already several steps away. So we're actually beginning to explore a STEM project with a elementary and middle school that actually already has a couple of MIT programs embedded in it. And what we're going to try and do is really understand by talking to 100 children how they define wellness and how we can potentially measure that objectively and think about, for this most common disease, how those basic ontologies are disrupted 50 years later in the patients that we already have. And so that type of in inverted commas, soft data, if we can harden it and objectify it, and more importantly, if we can collect it at scale, I think will help us understand what are the real elements of the biology that are important in the long run.
So one of the things I thought was fascinating is I think you have this boundless curiosity. And as all of us are thinking, like, what can I take away and what can I do? Like this never-ending quest of how do I learn everything I possibly can from this moment? And I hope I'm not violating a confidence, but I know that the way that you got introduced to this school where you're now studying what is wellness and how do children use language to define wellness is because it's the partner of the VC gentleman. And it's that constant seeking that I think is an extraordinary, that's how we will end up knitting together these, what we thought were disparate concepts.
Absolutely. I've actually done work with the school for about 6 years on and off, and one of the most amazing experiences was the first time we did a single cell to whole organism development, which you can do with zebrafish in 16 hours. And the very first question I had was from a second grader who asked me, what happens to fish when they die? And suddenly, as a biologist, you're explaining, not only death as a concept, but thinking about it not in a single dimension, but in so many different dimensions. Mm-hmm. And I think that's actually a critical part of healthcare. I think we err when we avoid thinking about what the patients want. Patients don't want a massive subclassification of their disease. They want to feel better or live longer. Mm-hmm. And if we keep that at the core, I think we're not straying too far.
Mm-hmm. Yes, I agree. I love this idea of translation.
Yes.
So you're working on how can you be translating in a way that allows your team that has all these different personalities and all these different areas of expertise to work together, right? So that means a translation from a language perspective. This translation from like how a child might describe something, but also the translation from idea all the way through to action that I know, Nancy, you think about all the time. Like, what are we doing? What are we doing? What are we actually doing to affect change? I want to give you one last quick quote as we're wrapping up, because I thought it stunned me. Everything, and comes back to translation and how the things that we think are complicated can be reduced down, and maybe in the reducing down is how we can build it back up to these systems. Everything we know about cardiovascular disease, we could have inferred from flywing genetics.
Yes.
Can you just describe to them what that means?
So the biology of the 6 genes that have been definitively implicated in coronary disease. 20 years ago, actually, one of my old collaborators won the Nobel Prize for understanding how these genes interact to form the wing of a fly. And from that information and the proteins that those proteins interact with, 20 years ago you could have inferred that the genetics of coronary disease would be the same or would overlap substantially with the genetics of diabetes, already known to be the case. That would also overlap with the genetics of blood pressure and ultimately also with the genetics of cognitive function. And those things have all come to pass to a greater or lesser extent, partly because of imperfections in the data then and the data now. But again, I think that's partly what we haven't done is bring both ends towards the middle. We haven't, I said at the start, discovery and clinical care are separate industries. Yeah. The only way you can really discover is by bringing these together and closing that data return cycle. And ultimately, that's what One Brave Idea is about. It's taking what we know already and maybe perfecting is not the word, but improving the amount of information content in what we already collect, and then adding new things to close that data return cycle and begin to build a truly learning system. And I think if we do that and bring everybody together around the same single problem, as we heard earlier tonight, and bring in industry and academia, data companies and social media, music and design, those things actually end up allowing us to identify what's wrong, change behaviors, move biology forward for the patient. And I think that ultimately is the goal we all have.
So, and what is your ask of us, right? This is a room full of folks who've got a whole different set of things that they're working on and skill sets. What can we be doing to help?
Telling us what you're doing that might be important to us, because we obviously, there's one of the biggest knowledge gaps is knowing what's out there, but also engaging very fully, being constructively critical of what we're doing. And one of the things we've already begun to do is reach out to the community now that we have data collected and start to ask people to come in and say, how does this fit? What are we missing here? How will we move this forward? That is actually an exciting thing to be able to do, simply because of the vision of the funders and the vision of the leadership group, and I believe the vision of my colleagues in being willing and able to move that forward, as well as the vision of the people who are willing to come in and kick the tires. So I'm looking forward to it.
So in closing, I want to share one other thing that you had said when I'd asked you that question before that builds on this. You said, I want you to feel like you're going to be empowered to be a part of the understanding of your own biology. and that's not a top-down thing, but it's a spontaneous thing from the bottom up, which I think for all of us, we don't see nearly enough of that in what we're doing. And I think it's such an extraordinary thing, what you're working on, your bravery and you're working on it, and a huge thank you to you guys for the support of the building of the center. I know as a mom and as a daughter, and as someone who has cardiovascular disease in my family, I'm so grateful that you guys are doing what you're doing. So thank you.
Thank you.